Welcome to the Creative Commons RX podcast. Before diving into this episode, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.
Welcome to the Deep Dive. If you've been watching the medical news, really, even just over the past few years, it's clear we're in the middle of a huge scientific shift, a real paradigm shift. For generations, medicine was mostly about managing chronic conditions, right?
Yeah.
Keeping symptoms under control, trying to extend life.
Standard practice, yeah. Symptom management.
But that focus is really changing now. We're moving towards these incredibly targeted interventions, even genetic modification potentially, and, the big promise, a one-time cure.
It really is more than just a change. It feels like a revolution. And it's being driven by, I'd say, three main classes of advanced therapeutics, all interconnected.
Okay.
You've got the big category of biologics, then the really cutting-edge stuff, cell and gene therapies or CGTs, and then something economically vital, biosimilars.
Right. And the second these breakthroughs, these amazing scientific leaps, leave the controlled environment of the lab, they run smack into, reality, systemic reality.
That's putting it mildly.
When you have a cure that might cost millions, literally millions of dollars, and involves changing a patient's basic biology, the ethical questions, the economic fallout, the sheer logistics, it's enormous.
It's a perfect storm of complexity.
So the mission for us today in this deep dive is, yes, to unpack the science. We need to understand that, but maybe more importantly, to really dissect the structural barriers, the things standing in the way.
Barriers around finance, access, social justice.
Exactly. These barriers threaten to make these potential miracle cures available only to, a very small select group.
And these issues are completely tangled together. The difficulty in just making these things, the unbelievable price tags, the ethical lines we're suddenly forced to draw, it all comes together to decide whether this whole revolution actually reaches people equitably, or if it doesn't.
Okay, let's unpack this then, because I think when the dust settles, the biggest challenge here isn't necessarily the science itself, though it's complex.
The science is incredible, but yeah.
It's the structure of our society, our healthcare systems, how we pay for things. So let's start at the beginning. Let's define what we're actually talking about on this new therapeutic frontier. So first up, biologics. This is kind of the foundation. People often mix these up with regular drugs, small molecule drugs, but they're fundamentally different. How should we picture a biologic? What makes it distinct?
Well, the key difference is the source. Unlike a typical small molecule drug, think aspirin, ibuprofen, something synthesized chemically, a biologic comes from a living system.
Living system? Like what?
That could be human cells, animal cells, or even microbial cells. It covers a really wide range of products, actually. Vaccines, blood components, but also these very large complex molecules like proteins or nucleic acids. In the U.S., the legal definition comes from Section 351 of the Public Health Service Act, just to ground it.
Okay, so the source material is alive or derived from something alive, and that dictates its complexity, right? A normal drug, small molecule, you can pretty much write down its chemical formula, make an exact copy anywhere.
Precisely. That's the critical distinction. Biologics are often huge molecules, incredibly intricate compared to small molecule drugs, because they come from living cells through complex processes like fermentation or cell culture. Well, you can't fully characterize them down to the last atom. It's just not possible.
Which means
there's always going to be a natural and, importantly, acceptable degree of batch-to-batch variability. Each batch produced is slightly different from the one before it, even if they work exactly the same way clinically. You simply cannot make a perfect, identical chemical copy like you can with a simple chemical drug.
And that basic fact, that structural reality that must dictate how you handle them too, creates logistical issues from the get-go.
Absolutely. Because they're often proteins, they're typically very sensitive to heat, so you need specialized handling, refrigeration, sometimes freezing, sometimes even cryogenic storage, which gets really expensive and complicated. And because they're so large, they usually can't be taken as a pill. They won't survive the stomach or get absorbed properly. So, administration almost always means injection or infusion, often in a clinic or hospital setting, not just something you pick up at the pharmacy.
Right. And when we talk about common biologics, what kind of treatments are we thinking of?
We're talking about major drug classes now. Things like monoclonal antibodies, that's a huge area, including drugs like infliximab for autoimmune conditions, also cytokines, certain growth factors, treatments that have already transformed care for many diseases.
Okay, so that sets the stage for the real game changers, the therapies that grab the headlines. Cell and gene therapies, CGTs, these feel fundamentally different because they're working at the level of our DNA.
Yeah, CGTs are really the ultimate in targeted medicine. The whole idea, the mechanism of action, is to fix the underlying genetic problem causing the disease. We usually talk about three main ways they do this. One is replacing a gene that's faulty or missing with a working copy. Another is inactivating a gene that's malfunctioning and causing harm. And the third is introducing a totally new gene, or maybe a modified gene, to give the body a new ability, perhaps to fight off cancer cells, for example.
And delivering these genetic instructions, that sounds pretty revolutionary too. How did that actually happen? How do you get the gene into the cells?
There are two main strategies. The first is called in vivo therapy. That's Latin, in the body. Basically, the genetic material often packaged inside a harmless virus, a viral vector that acts like a delivery truck, is given directly to the patient, maybe through an infusion. The patient's own cells then take up this new genetic material.
Okay, direct delivery. What's the other way?
The other way is ex vivo, meaning out of the body. This is often more complex, definitely more personalized. Here, you actually remove cells from the patient, take blood, for instance. Then those cells are sent to a specialized lab, genetically modified there, often grown or expanded to get more of them, and then finally reinfused back into the same patient.
Wow. That ex vivo process sounds incredibly intensive. Tailor-made, almost. Is that the science behind those famous CAR-T cancer therapies we hear about?
Exactly. CAR-T therapies like Kymriah, Yescarta, they were real pioneers. They work by taking a patient's own immune cells, their T cells, engineering them ex vivo to recognize and attack their specific cancer, and then putting them back in. It's revolutionized treatment for certain blood cancers.
Amazing.
And beyond cancer, we're seeing gene replacement therapies making huge strides in rare genetic diseases. Think Zolgensma for spinal muscular atrophy SMA, which used to be a leading cause of death in infants. Or, more recently, the gene editing therapies like Casgevy for sickle cell disease. These are potentially curative. We're not just managing symptoms, we're talking about fixing the root cause.
So the complexity of biologics, the fact you can't make an exact copy, that leads us to the third category you mentioned, biosimilars. How do you get generic-like competition if you can't make a generic?
Right, you can't make a true generic. That's the absolute key point, legally and scientifically. A generic drug has to be chemically identical. A biosimilar, on the other hand, is defined as a biologic product that regulators determine is highly similar to an already approved biologic, the reference product.
Highly similar.
Yeah.
But not identical.
Correct. And critically, there must be no clinically meaningful differences in terms of safety, purity, or potency. Because you can't prove identical structure, regulatory agencies like the FDA demand what's called a totality of the evidence approach, meaning they look at everything. Detailed analytical chemistry comparing the molecules, animal studies, and usually human clinical trials, all comparing the proposed biosimilar directly against the original reference biologic. It's a very rigorous process.
So it's proven to work the same way, be just as safe, even if it's not molecule for molecule identical. Got it. And what's the key mechanism that lets these biosimilars actually bring down costs effectively?
That comes down to a special designation called an interchangeable biosimilar. This requires even more evidence submitted to the FDA, often including studies showing you can switch patients back and forth between the original and the biosimilar without any issues.
And if it gets that designation?
If a biosimilar is deemed interchangeable, then depending on state pharmacy laws, a pharmacist can substitute it for the original prescribed biologic without needing to call the doctor first, just like they do with regular generics.
Ah, okay, so that's the fast track to savings.
It's the most powerful tool we have for driving competition in the biologics market. It increases patient access, lowers costs, and this is really important. It creates fiscal headroom. It frees up money in the healthcare
The
system.
money that we desperately need to afford the next wave of innovation, like those multi-million-dollar gene therapies.
Precisely. Biosimilars are crucial for the sustainability of the whole system.
And that fiscal headroom couldn't be more critical, because we're now talking about costs that just, well, they break the old models of healthcare economics. The price tags for some of these new cures are genuinely hard to fathom.
They really are. It's a massive economic shock to the system. We're seeing treatments, often just a single infusion, priced in the millions. Libmeldy, for a rare disease called MLD, is listed at $4.25 million.
$4.25 million.
Hemgenix for hemophilia B is $3.5 million. Even the CAR T therapies, the drug itself, might be, say, $373,000 to $475,000, which is already huge. But then you add the long hospital stay, the specialized care, managing side effects, the total cost of care frequently goes over $1 million per patient.
$1 million?
Yeah.
For one person's treatment. We have to understand what's driving that. It can't just be profit-taking, right? Though that's often the narrative. There must be some underlying economic factors specific to this new kind of medicine.
You're right, it's complex. There are probably three main drivers, maybe more, but three big ones. First is the sheer cost of R&D and the manufacturing. Bringing one of these novel therapies to market can easily cost upwards of $5 billion.
$5 billion. Okay.
But the manufacturing is the really unique part. It's not like stamping out pills on an assembly line. Many CGTs, especially the ex vivo ones like CRRT, are autologous. That means they're made using the patient's own cells.
So each batch is custom made for one person.
Exactly. It's incredibly complex, often manual work, requires highly sterile clean room environments, super skilled technicians. You're essentially commissioning a bespoke, handcrafted therapy for an individual. There are basically no economies of scale. The fixed costs per treatment remain incredibly high.
That handcrafted analogy, yeah, that makes sense. It's the total opposite of mass production efficiency.
Totally. The second major driver is small patient populations. These therapies often target rare, sometimes ultra-rare, diseases, conditions that might only affect a few thousand or even just a few hundred people worldwide.
Right. So that huge $5 billion R&D cost
Has to be recouped from a very, very small number of patients. The denominator in that equation is tiny, which pushes the price per patient way up.
Okay, that makes sense too. And the third driver is how manufacturers justify the price, this idea of value-based pricing, the argument that it saves money in the long run.
That's the theory, yes. And this is where it gets really contentious between manufacturers and payers, like insurance companies. The argument is, okay, $3 million today sounds like a lot, but it's cheaper than paying, say, $300,000 every year for 30 years for hospital visits, ongoing drugs, lost productivity.
Offsetting a lifetime of chronic care costs.
Exactly. But payers push back hard on this value argument for a couple of key reasons. First, the durability is often unknown. These treatments are so new. Payers are being asked to fork over millions upfront based largely on clinical trial data and predictive models. They often don't have solid real-world proof that the cure will last for 10, 20, 30 years. What if it wears off after 5?
That's a huge gamble for the payer.
It is. And the second big issue is specific to the, let's say, fragmented nature of the U.S. insurance market.
Right. People change jobs, change insurance all the time.
Correct. Patient mobility is high. So the insurance company that pays that massive multi-million-dollar upfront cost for the gene therapy often isn't the same company that will actually see the financial savings 10 or 15 years down the road when the patient doesn't need chronic care anymore.
The payer pays the cost, but someone else potentially reaps the benefit later. A total misalignment of incentives.
Exactly. It creates this huge disincentive for the initial payer to quickly approve and cover these incredibly expensive treatments, even if they represent great value for the patient and for society overall.
So given that massive upfront cost, which, as you say, is hard for any single payer to swallow in one budget year, how do we make this sustainable? We know biosimilars help with older biologics, but for these brand new multi-million-dollar CGTs, what are the solutions?
Yeah, we absolutely have to move beyond the old fee-for-service model. It just doesn't work here. For CBTs, we're seeing several innovative payment models emerge, designed to manage both the clinical risk and the financial shock.
Okay, like what?
The main one people talk about is the outcomes-based agreement, or OBA, sometimes called pay for performance.
So payment depends on whether the treatment actually works.
Essentially, yes. It shifts some of the clinical risk from the payer back to the manufacturer. The agreement specifies certain clinical milestones. Maybe the patient needs to remain free of symptoms or avoid hospitalization for a set period, say 3 or 5 years.
And if they don't meet those milestones?
If the therapy doesn't deliver the promised outcome, the manufacturer has to pay a rebate or refund part of the cost. It tries to tie the payment directly to the demonstrated long-term value, not just paying when the drug is infused.
That sounds sensible. A way to handle that durability uncertainty problem. But what about the immediate sticker shock, paying $4 million now?
That's where other models come in, like amortization or installment payments, basically spreading the cost out over several years. Instead of one massive hit to the budget, the payer makes smaller, more manageable payments over, say, 5 years.
Like financing a really, really expensive car.
Kind of, yeah. It smooths out the budget impact significantly. But this brings us right back to that problem of patient portability.
If the patient changes insurance in year 3 of a 5-year payment plan.
Exactly. Who is responsible for the last 2 years of payments? The old insurer,
Yeah.
the new insurer. Does
Yeah.
the manufacturer chase the patient? It gets incredibly complicated logistically and contractually, especially with competing insurance companies. That complexity is definitely slowing down the adoption of these models.
Sounds like an administrative nightmare.
Hmm.
Any other approaches?
Well, there's also risk spreading or reinsurance. This is particularly relevant for smaller insurance plans or state Medicaid programs. They might pool their risk together or buy specific reinsurance policies, basically insurance for the insurer.
To protect themselves if they suddenly get hit with several multi-million-dollar therapy claims in 1 year?
Precisely. It prevents 1 or 2 catastrophic cases from bankrupting the plan. So combinations of these models, OBAs, installments, risk pooling, are really essential if these cures are going to become accessible beyond just a handful of patients.
Okay, so we've talked money, we've talked access in terms of cost. Now let's pivot to perhaps the most profound ethical boundary these technologies force us to confront, the potential to edit the human genome itself. And this whole huge debate really hinges on one fundamental distinction, doesn't it?
It absolutely does. It's the difference between somatic gene editing and what's called heritable human genome editing, or HHGE. And this is a line that globally society has been extremely hesitant to cross for very good reasons.
Well, let's clarify first what is currently happening, what's ethically accepted in the therapies available now.
Right. All the approved gene therapies we have today use somatic gene editing. That means they target non-reproductive cells in the body, somatic cells. Think blood cells, liver cells, muscle cells, neurons. The genetic modifications are confined entirely to that individual patient receiving the treatment. And crucially, these changes cannot be inherited. The patient's children won't carry the genetic modification.
So somatic editing treats the individual, but HHGE, that's about editing the future.
That's the heart of it. Heritable human genome editing involves modifying the DNA of reproductive cells, sperm or eggs, or editing the DNA of an embryo at a very early stage. These changes become part of every single cell in the body of the resulting person. And because they're in the germ cells, they are heritable. They get passed down to all subsequent generations, forever.
Wow, that raises the stakes astronomically. And in the US, there's basically a block on doing HHGE clinically, right? Though it's maybe not a straightforward law.
That's correct. It's what we call a de facto moratorium. It's not a specific law saying thou shalt not edit the human germline. Instead, it's done through funding restrictions passed by Congress year after year.
How does that work?
Congress includes language in appropriation bills that prohibits federal funds specifically for agencies like the FDA or the National Institutes of Health, NIH, from being used to review or support any research involving the intentional genetic modification of human embryos that would be heritable. So no government money, no regulatory review pathway. It effectively stops any clinical trials from moving forward in the U.S.
Okay, so why is there such widespread concern, almost alarm, about opening that door to HHGE? What are the main arguments against ever allowing it?
The number one concern is safety and the risk of irreversible consequences. Even our best gene editing tools like CRISPR aren't perfect yet. There's always a risk of off-target edits, unintended changes elsewhere in the genome's 3 billion letters. And there's also the risk of mosaicism, where the edit doesn't happen uniformly in all cells, leading to a mix. Now, if that happens in somatic editing, it affects one person. If it happens in HHG, that error is permanent. It gets locked into the human gene pool. You could inadvertently create a new heritable disease that you can't recall. It's an experiment on the entire human species with potentially devastating irreversible outcomes.
And the whole idea of consent just breaks down completely.
Fundamentally, the ethical principle of informed consent is bedrock in medicine. But you simply cannot get consent from the person most affected the embryo who will become the child, or from all the future generations who will inherit that altered DNA. Critics argue that making such fundamental, permanent changes to someone's genetic makeup without their consent is a profound violation of their future autonomy, their right to inherit a genome that hasn't been intentionally designed by others.
Then there's the fear. Everyone talks about the slippery slope.
Yes, the slippery slope argument is pervasive. It's the societal worry that if we allow HHG, even for clear therapeutic uses, say, correcting a gene that causes a terrible disease like Huntington's, it will inevitably, eventually, lead to its use for non-therapeutic enhancement.
Designing babies.
Pretty much. Once the technology is safe and normalized, the fear is the pressure will grow to use it to optimize children, enhance intelligence, physical appearance, athletic ability. It blurs that critical line between healing disease and manufacturing better humans, which raises deep questions about what it means to be human, about diversity, about accepting imperfection.
And tying back to our earlier discussion, if these HHGE techniques were developed and were incredibly expensive, like CGTs are now, the justice implications are huge.
Oh, absolutely. If HHG is only available to the wealthy, it could create a literal genetic divide, a future where the rich can afford to give their children genetic advantages while the poor cannot. This wouldn't just exacerbate existing inequalities, it could potentially lock them into our biology, creating a permanent heritable class system written in DNA.
It's a dystopian scenario. Now, despite all these very strong arguments against it, are there any proponents who argue for HHGE, maybe in very limited cases?
There are some arguments, usually centered on beneficence in extremely rare situations. For example, imagine a couple where both partners carry genes for a devastating dominant disease, or both are homozygous for a severe recessive condition like Tay-Sachs. Maybe establish options like pre-implantation, genetic diagnosis, PGD testing embryos before implantation aren't feasible or effective for them for some reason.
So in those niche cases,
Proponents might argue that HHGE could be the only way to ensure they have a healthy biologically related child, preventing certain and severe suffering. It's framed as a last resort for profound medical need.
what did the big U.S. scientific bodies like the National Academies say about this? Do they recommend a permanent ban?
The National Academies of Sciences, Engineering, and Medicine, NASM, put out a major report in 2017. They took a very cautious stance. They did not call for a permanent, absolute ban forever. However, they said clinical trials for HHGE should only potentially be permitted in the future under incredibly strict conditions.
Such as?
It must be aimed at preventing a serious disease, there must be no reasonable alternatives, there needs to be credible preclinical data showing it's likely safe and effective, and this is key. There must be plans for rigorous, long-term, multi-generational follow-up to monitor the consequences. Essentially, they set the bar extremely high, acknowledging the profound risks and ethical concerns. The burden of proof for safety and necessity would have to be extraordinary.
Okay, so we've covered the immense cost, the complex payment models, the deep ethical waters of germline editing. Let's bring it back to the practical reality for a patient today. Even if the cost hurdle is somehow cleared, say insurance agrees to pay, there are still major structural and logistical barriers that can stop someone from getting treatment, right?
Absolutely. The sheer logistical complexity of delivering many CGTs is a huge barrier in itself. These therapies usually aren't available at your local hospital. They're restricted to a relatively small number of highly specialized academic medical centers, designated as Qualified Treatment Centers, or QTCs.
Why only those centers?
Because you need the infrastructure, the specialized staff trained in handling these agents, the ultra-low temperature or cryogenic storage facilities, the intensive care units capable of managing potentially severe side effects. It's a high bar.
Which immediately creates a problem if you don't live near one of those major centers.
Exactly. It leads to significant geographic disparities. Researchers actually talk about CGT deserts, large parts of the country where patients might have to travel hundreds, even thousands of miles just for a consultation, let alone for the weeks or months required for the actual treatment and follow-up.
Wow. And the supply chain itself sounds fragile.
Incredibly fragile, especially for the ex vivo therapies. You have the initial cell collection, leukapheresis. Then those cells are shipped, often cryopreserved, to a centralized manufacturing facility, maybe only one or two in the whole country or continent. They're modified there, then shipped back, again under strict cold chain conditions, to the QTC for infusion. Any delay, any temperature fluctuation, any damage in transit.
And the whole personalized batch, that multi-million-dollar treatment, could be lost.
Precisely. The patient might have to start the whole process over if they're even still well enough to do so.
Okay, so let's say a patient overcomes the travel, the logistics. They arrive at the QTC. They still face another massive wall, don't they? The insurance company's approval process.
Ah, yes, the administrative barriers. This is often the final, frustrating choke point. Payers, understandably given the cost, impose extremely stringent prior authorization requirements. We're talking mountains of paperwork, sometimes over 100 pages of documentation needed.
100 pages?
Easily. And payers often apply very narrow clinical criteria for approval, sometimes even stricter than what the FDA approved the drug for. This creates a huge burden for the doctors and their staff, and more importantly, it leads to delays. Weeks, sometimes months, waiting for approval.
And for someone with a fast-moving cancer or a rapidly progressing rare disease, those delays can be devastating.
They can literally be the difference between being eligible for a potentially curative therapy and the disease progressing to a point where they're no longer a candidate. It's heartbreaking when administration gets in the way of science.
This leads directly into the impact of what we call the social determinants of health, SDOH. Because even if the drug cost is covered, the cost of accessing the treatment often isn't.
That's a critical point. Financial toxicity is a huge issue. A patient's overall economic stability plays a massive role. It's not just about affording potential copays or deductibles, which themselves can be tens of thousands of dollars for these drugs.
Right, it's everything else.
It's all the indirect costs. The patient, and often a caregiver, might need to temporarily relocate near the QTC for weeks or even months. Think about the cost of travel, gas, flights maybe. Lodging, finding affordable accommodation near a major medical center isn't easy. Childcare back home. And crucially, lost wages. Both the patient and their caregiver likely have to take significant time off work. For many families, especially low-income families, these indirect costs alone make the treatment completely inaccessible, even if the drug itself is technically covered.
And the process itself demands a lot from the patient, doesn't it, in terms of understanding everything?
Definitely. It requires a high degree of health literacy. Patients need to be able to understand complex information about the therapy, the difference between somatic and germline perhaps, the potential for serious side effects like cytokine release syndrome we mentioned earlier. They have to weigh really complex risks and benefits, navigate insurance appeals, manage appointments.
It's overwhelming.
It is. And patients with lower levels of education or those facing language barriers, they inevitably struggle more to navigate this complex system successfully. This, again, tends to concentrate these advanced therapies among more affluent, highly educated populations.
And do we see evidence that these pressures, geography, cost, complexity, lead to actual racial and ethnic inequities in who gets these treatments?
Sadly, yes. Studies consistently show disparities. Even when you control for factors like having insurance, minority patients are often less likely to receive these advanced biologics or be referred for CGT evaluation.
Why is that?
It's likely a combination of factors. Being underinsured plays a role, the disproportionate impact of those indirect costs and financial toxicity we just discussed. There can also be issues with trust in the healthcare system, communication barriers, and potentially implicit bias in referral patterns. Maybe primary care doctors or community specialists are less likely to refer minority patients to the big academic QTCs. The access desert isn't just geographic, it's often socioeconomic, existing right within diverse cities, just blocks away from the treatment centers.
It's a systemic problem layered on top of the science. Now let's shift slightly to the ethics of the information itself. When you do the kind of deep genetic sequencing needed for some of these therapies, or just genetic testing in general, you often find things you weren't looking for, right? Actionable information unrelated to the main reason for the test.
That's right. This is the challenge of ancillary genetic findings, or incidental findings, and it creates a real ethical tension for clinicians. On one hand, you have the duty to warn the principle of beneficence, wanting to tell the patient about a preventable risk you've discovered. But on the other hand, you have the patient's right not to know their autonomy, their right to decide what health information they want about themselves.
So how does medicine handle that? You find something serious but unrelated, do you just tell them?
It's managed carefully. Professional groups like the American College of Medical Genetics and Genomics, ACMG, have thought a lot about this. They distinguish between just any random finding and what they call actionable secondary finding,
Actionable meaning.
meaning findings in specific genes that are known to significantly increase the risk of a serious disease, but where there are effective preventative measures or treatments available. The ACMG actually recommends that labs actively look for and report findings in a specific list of these genes. I think the list is up to 73 genes now associated with conditions like certain inherited cancer syndromes or heart conditions.
So the default is towards disclosure for these high-risk, actionable conditions. But the patient still has control.
Absolutely. Patient autonomy is paramount here. The crucial step is pre-test counseling. Before any genetic testing happens, the patient must be clearly informed that these kinds of secondary findings might be discovered. And most importantly, they must be offered the choice to opt out of receiving this secondary analysis. They can say, yes, do the main test, but I do not want to know about any unrelated risks you might find.
So they retain the right not to know.
Exactly, because once that information about a future risk is disclosed, you can't unknow it. It changes your life, your planning, your anxieties, so the patient's right to refuse that knowledge has to be respected.
But that autonomy, it runs into a major potential problem, a big gap in legal protection, doesn't it, regarding genetic discrimination?
This is probably one of the biggest practical concerns patients have when deciding whether to opt in to receiving secondary findings. There's a key piece of U.S. federal law, the Genetic Information Non-Discrimination Act of 2008, usually called GINA.
And GINA protects people.
Yes, it provides really important protections. GINA prohibits health insurers from using your genetic information to deny you coverage or charge you higher premiums. It also stops employers from using genetic information in hiring, firing, or promotion decisions. That's huge.
But there's a but.
There's a critical cap.
Yeah.
GINA's protections do not extend to other types of insurance, specifically life insurance, disability insurance, and long-term care insurance.
Oh, okay. So
If you choose to receive an ancillary genetic finding, say you learn you have a BRCA mutation significantly increasing your risk of breast and ovarian cancer, even though the original test was for something else, that information, once it's in your medical record, could potentially be accessed by underwriters for these other types of insurance. They could legally use it to deny you a policy or charge you drastically higher rates.
that's a massive disincentive to learn potentially life-saving information.
It creates an agonizing choice for patients. Do I want this knowledge that could help me prevent a serious disease, but risk my ability to get affordable life insurance to protect my family, or disability insurance if I get sick? It's a terrible trade-off forced by this legal loophole.
Given everything we've discussed, the incredible operational complexity, the cryogenic handling, the bespoke manufacturing, navigating these multi-million-dollar payments and prior authorizations, it seems like the role of the pharmacist, especially in these specialized centers, must have evolved dramatically. They're not just dispensing pills anymore.
Yeah. Oh, absolutely not. They've become central figures, really indispensable stewards of this entire complex process. They are critical in operationalizing complexity within the QTCs. Pharmacists are often the ones developing, implementing, and enforcing the detailed institutional policies and procedures for the entire medication use system for CGTs.
Which includes
Everything from managing the intricate logistics of receiving, thawing, and preparing these therapies under strict timelines, dealing with that cryogenic storage, ensuring all the biosafety precautions are meticulously followed during handling. It's highly specialized work.
And clinically, they're involved in managing the risks too, like that cytokine release syndrome you mentioned.
Definitely. Pharmacists play a key role in monitoring patients for and managing the unique and potentially severe acute toxicities associated with therapies like cytokine release syndrome, CRS, is a big one. It's a systemic inflammatory response that can range from mild flu-like symptoms to life-threatening organ failure. Pharmacists help develop the protocols for recognizing it early, grading its severity, and guiding the use of specialized medications, like immunomodulators, to treat it effectively. They are really the medication safety experts on the front lines.
So beyond the direct clinical and operational roles, they're also involved in the financial and access side of things within the hospital.
Hugely important role there. They are central to financial navigation and advocacy. Pharmacists, particularly those in leadership or specialized roles, are usually deeply involved in the hospital's Pharmacy and Therapeutics P&T committee.
Remind us what the P&T committee does.
The P&T committee is the group within the hospital that decides which drugs get added to the hospital's formulary, the list of approved medications the hospital will use and stock. Pharmacists lead the evidence-based review of new drugs, including these high-cost therapies. They analyze the clinical data, the safety profile, and critically, they perform budget impact analyses to forecast how bringing in a multi-million-dollar drug will affect the hospital's finances. They're essentially the internal gatekeepers advising the institution on affordability and value.
But they also work directly to help patients overcome those cost barriers we talked about.
Yes, and this is arguably one of their most vital roles in promoting equity. Pharmacists or specialized pharmacy technicians under their supervision often act as expert financial navigators for patients. They are the ones who know the ins and outs of manufacturer copay assistance programs, grants available from charitable foundations, and other patient assistance programs.
So they actively help patients find money.
Exactly. They work proactively to connect patients with these resources, helping them piece together funding to cover copays, deductibles, sometimes even travel and lodging costs. They work to dismantle those financial barriers, fighting that financial toxicity head-on to ensure that cost doesn't prevent an eligible patient from getting the therapy. It's a critical advocacy function.
And finally, looping back to biosimilars, you said they're key to creating fiscal headroom. What's the pharmacist's role in making sure those savings actually happen?
They have a huge educational imperative when it comes to biosimilars. The uptake of biosimilars really depends on building trust from doctors and trust from patients. Pharmacists are often the primary educators. They're the ones explaining the science, the rigorous regulatory pathway, the difference between a biosimilar and an interchangeable biosimilar to both prescribers and patients.
And that involves tackling fears or misconceptions too, right?
Absolutely. They need to address patient anxieties. Sometimes you see a nocebo effect, patients assuming a lower-cost medicine must be less effective or have more side effects, even when the data shows it's equivalent. Pharmacists counsel patients, provide reassurance based on evidence, and advocate within the institution for policies that promote evidence-based substitution, especially using interchangeable biosimilars where appropriate. By driving biosimilar adoption, they directly help the health system realize the significant cost savings.
Savings that can then be reinvested into funding access to the next wave of genuinely innovative, potentially curative therapies.
Yeah.
It's all connected.
It's all deeply interconnected.
So wrapping this up, this deep dive really highlights a stark contrast, doesn't it? We're standing at this peak of incredible scientific achievement, the power to potentially cure diseases that were death sentences just years ago. And yet our systems, our ethical frameworks, our economic models, our delivery structures are lagging behind, struggling to cope.
It's a fundamental tension. The innovation is racing ahead, but our ability to deliver it equitably is challenged.
And it forces us to ask that hard question. If an innovation, no matter how brilliant, is only realistically available to the wealthiest or the best connected, is it truly a societal advancement? Or does it risk deepening the divides we already have?
The path forward really requires a systemic approach, not just a scientific one. To make sure these breakthroughs benefit everyone, we need to consciously build equity into the process. That means things like prioritizing shared decision-making, making sure patients are truly informed partners in these complex choices. It means moving towards a more holistic value assessment, looking beyond simple cost per QA metrics to really consider the severity of the disease, the unmet need, the impact on families and society. And maybe most importantly, it means doing proactive equity audits, really examining every step from who gets enrolled in clinical trials to how prior authorization criteria are set to where treatment centers are located to find and dismantle those structural barriers that perpetuate disparities.
The ultimate measure. It won't just be about the diseases we conquer, will it?
I don't think so. If we connect this to the bigger picture, the ultimate measure of these remarkable scientific achievements will not be the diseases we can conquer, but whether we do so in a way that affirms our shared commitment to a just, equitable, and compassionate society. And the science isn't stopping. We're seeing new frontiers emerge in vivo gene editing becoming more feasible, even speculation about technologies like artificial wombs. These will raise even newer, perhaps even more complex ethical questions.
So the questions we grapple with today about access, about cost, about ethics, they're just the beginning. They set the stage for how we'll handle the even more profound choices science will undoubtedly present us with tomorrow.
That seems right.
So here's the thought to leave you with. When the cure costs millions and the science itself demands or at least reveals intimate details about our very DNA, who gets to decide? Who decides who gets access to potentially life-changing treatment? And perhaps more fundamentally, what parts of our shared humanity, fairness, compassion, equity do we risk eroding if we prioritize efficiency or profit, or even just scientific progress itself over ensuring that progress benefits everyone? Something to really think about as we all navigate this incredible and incredibly complex new era of medicine. Thank you for joining us for the deep dive.
This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.